Hi guys, welcome to HSC series. Now today we're going to start off with bio with the first focus, maintaining a balance. So there's really a few core things to think about in this focus, which we will be going on through this series. Basically, it's just talking about how organisms create a stable environment and how that affects the functioning of their cells and more broadly the functioning of the organism. So what we want to outline is what is the stable environment. So we'll be looking mostly at humans, so we're talking about internal temperature, substrate concentration, enzymes in the body, and how they function, such as controlling the endocrine system and nervous system, and how this affects the overall organism. And then we want to focus on the mechanisms that keep this stable environment, so feedback systems and receptors and effectors. And then we want some specific examples as to how enzymes affect organisms. Now, it's really important that we use specific examples because that's what they're asking about in the syllabus. So we will do both humans and plants and animals as examples. And also to note, in each of the bio focuses, there'll be first and second-hand investigations, which are crucially important because they ask them mostly on long responses. So we'll go through those later. Okay, so the first dot point in the maintaining balance focus is all about enzymes. So describing their role, their chemical composition, and a model which explains their specificity. So first things first, we've got to outline what are enzymes. So enzymes are biological catalysts which increase the rate of chemical reactions within the metabolism. So what's the metabolism? The metabolism is all chemical reactions occurring within living organisms. So basically, enzymes speed up all the reactions occurring within an organism, and they do this by influencing the stability of bonds within cells. So less energy is required to break these bonds, and therefore reactions can occur faster. Yeah, so just outlining that reactions are part of the metabolism. So this is crucial to the functioning of cells and organisms. And then we're next asked, what is the chemical composition of enzymes? So enzymes are made up of large proteins. Oh, it's important to note that enzymes are not used up in this process, so they can be used on and on and on. Yeah, so enzymes are made of large proteins, one or more polypeptide chains, and obviously that's made up of amino acids, and they form a globular shape, so they're not really a very like straight shape, they're kind of roundish, 3D. Now we're up to describing the model that shows their specificity. So basically the first one that was introduced is the lock and key model, which explains how enzymes' active sites fit perfectly with specific substrates. So only a specific substrate will fit in with a specific enzyme. Now that model is helped to produce the one that's used today, which is the induced fit model, which shows how enzymes' active sites fit specifically with substrates, but upon that fitting together, the enzyme actually molds its active site to fit even further.